Assalamu alaikum. Welcome to my lecture. So, in this lecture, we'll discuss about the primary structure of the protein. In the last lecture, we previously described the different levels of the protein organization, different structures of the proteins. So, proteins usually have very complex structures. That's exactly why we normally break down the complexity into the different levels. So we have here the simplest level of the complexity, that is the primary structure, and then we'll go all along the, you can say all the way up to the highest level of the complexity, that is from the primary structure to the secondary, tertiary, and ultimately to the quaternary structure of the protein. So, what do we mean by that? That this is the primary structure. So, this primary structure of the protein basically describes the sequence of the amino acids in the polypeptide chain. So you can say that every protein in our body that is made up of the unique sequence of the amino acid in the polypeptide chain. Now it turns out that you can say that every single protein that is present inside our body has its own unique sequence of the amino acids, and it is its primary structure, that is the sequence of the amino acids, that actually determines its final form, its final, you can say its final 3D structure of the protein. So what do we mean by this primary structure? Primary structure is basically the unique sequence, linear sequence of the amino acids in the polypeptide chain that determines its final three-dimensional structure. So let's suppose we have this linear chain of the amino acids. So here you can see there are five amino acids. Amino acid number one, amino acid number two, three, four, and five. These five amino acids are basically linked together by the peptide bonds. Here you can see these are the peptide bonds that are shown in green here. There are, you can see there are four peptide bonds here, as this peptide bond, one, two, three, and four. Four peptide bond here, and we have five amino acids here. So this is, you can say, a polypeptide chain having five amino acids and four polypeptides. So this linear chain, you can see it has polarity. What do we mean by that? Here you can see that first of all, every amino acid within this polypeptide chain is referred to as a residue. जो हर amino acid होता है, individual amino acid that is making the polypeptide chain, उसको हम क्या कह रहे हैं? That is known as the residue. Amino acid को हम क्या कह रहे हैं? It's the residue. So here we have five amino acid residues, but we have four peptide bonds. So we can say that if we have n number of the amino acids, then their number of peptide bonds will always be n minus 1. If we have 6 amino acids, then the peptide bonds are going to be the 5. If we have 9 amino acids, peptide bonds are going to be the 8. Similarly, we have 13, 14, 15 amino acids, and then peptide bonds will have 12, 13, and 14. इस जितने भी अमीनो एसिड्स होंगे हमारे पॉलीपेप्टाइड चेन से, उससे एक नंबर कम will be our number of the peptide bonds. Now, under normal physiological conditions, at a what do we mean by that? At a neutral pH, this polypeptide chain here, you can see that this alpha amino group will have a full positive charge here, and at the end of this chain, this carboxyl group is having a full negative charge here. So we have a, you can see we have a polarity, that is result of these two different charges. That is the positive charge on this side, on the left side on the amino group, and the negative charge on the right side on this carboxyl group. So by convention, what do we see? That the beginning of the polypeptide chain will always set to be at the positive end. कि जब भी polypeptide chain का start होगा, that is always from its positive side. ठीक है? And its ending is always set to be at its negative sides. That is the beginning from the alpha amino group and the alpha amino side, and the ending will be at the alpha carboxy side. Now, notice if we go along a polypeptide chain, Now, what do we see here? We see here that these repeating units here, these can be NCC. Again, we have NCC. This again we have NCC. So these are the constant repeating units all over the polypeptide chain. So if this can be continue if we continuously add other further amino acids to this polypeptide chain. So what do we say that the this each repeating units, these repeating units, or you can say these repeating sections in the polypeptide chain, this is known as the backbone. इसको हम क्या कह रहे हैं? These are known as the backbone. So in this case, you can see this NCC. Again, we have peptide bond, then we have NCC. Again, we have NCC. This is constant section of the amino acid that is forming the backbone of the polypeptide chain. So this is the constant section, constant repeating unit all over the chain. So this is the known as the backbone of the polypeptide chain. Now we have a variable section in the polypeptide chain. This variable portion of the polypeptide chain, that is because of these R groups. Here you can see that this is the alpha carbon. So alpha carbon pe we have R group. In this amino acid number 1 we have R group 1. Amino acid number 2 we have R group 2. And this is the amino acid number 3. Here we have R group 3. So there are 5 amino acids having the constant backbone. इनकी constant backbone है, that is the NCC. And with the different groups, these are basically the R group, R1, R2, R3. These are the side chain groups. Okay, so we can see that there are two sections in our polypeptide chain. One is the repeating units, that is known as the backbone, and other one is the variable section, variable portion, that are because of these side chains. Now here you can see, let's discuss that this whole polypeptide chain basically has a great potential to form the hydrogen bonds with the other atoms or the molecules. So that's why there will be the formation of the other structures of the protein that lead to their secondary structure in the formation of the alpha helix as well as the beta pleated sheets. जिसपे there is the formation of the hydrogen bonds. So that's because why this polypeptide chain has a great potential to form the hydrogen bond. This is because of these two groups here. Here you can see this NH group and this is C double bond O, that is the carbonyl group. So these two groups basically act as a donor and the hydrogen bond acceptor group. Here you can see this N and H group. This basically act as the hydrogen bond donor group. ठीक है? So this bond is the hydrogen bond donor group and this bond is the hydrogen bond acceptor group. So here you can see that this nitrogen atom is basically the more electronegative as compared to the hydrogen. So this nitrogen also have a lone pair of electrons. So it is more electronegative as compared to the hydrogen atom. So whenever more electronegative atom in the will come in close proximity to the hydrogen atom, as this hydrogen atom basically is carrying the partial positive charge. क्योंकि अगर नाइट्रोजन ज्यादा इलेक्ट्रोनेगेटिव है तो नाइट्रोजन will carry the partial negative charge and the hydrogen will carry the partial positive charge. So what we ensure that if we have some other atom that is electronegative in the close proximity, then in that case this will be donated to the that particular electronegative atom. So we are going to form an hydrogen bond because the hydrogen bond is going to form. So this group basically act as the hydrogen bond donor. ठीक हो गया? So what do we say that this nitrogen, this NH bond basically act as a hydrogen bond donor because nitrogen is more electronegative as compared to the hydrogen. This nitrogen carrying the partial negative charge, this hydrogen carrying the partial positive charge. So अगर कोई भी एटम जो कि हाइड्रोजन से ज्यादा इलेक्ट्रोनेगेटिव है वो इसकी क्लोज प्रॉक्सिमिटी में आएगा, देन ये क्या करेगा, ये हाइड्रोजन एटम उसके विल एक्ट एज अ डोनर फॉर दैट इलेक्ट्रोनेगेटिव एटम और वो क्या बना लेगा, हाइड्रोजन बॉन्ड बना लेगा. सो सिमिलरली we have uh, so here we have the another group, that is the hydrogen bond acceptor group. Here you can see this C double bond O. Here the oxygen is basically the more electronegative than the carbon, so it develops a partial negative charge. And so if any hydrogen atom that is carrying the partial positive charge is found in the close proximity on the other side of this, so this partially negative oxygen, then we have a bond that is formed, that is the hydrogen bond is going to form between this oxygen atom and the partial positively charged hydrogen atom. So every single what can we say, that the every single NH bond and the C double bond O, this carbonyl bond in polypeptide chain, will act as a hydrogen bond donor as well as the hydrogen bond acceptor molecule. That's why this polypeptide chain basically has a great potential to form the hydrogen bonds with the other atoms or the molecule. Now, let what do we see here, that these different amino acid 1, 2, 3, 4, these are linked together through these amide linkages. These are the peptide bonds. So let's discuss the nature of the peptide bond. So basically this nature of the peptide bond is, you can say this peptide bond is apparently looks like the single bond, but it has a double bond character. Okay, this basically peptide bond is having the double bond character. This double bond character क्यों है इसका? This double bond character is basically because of the resonance stabilized. So ये क्योंकि resonance stabilized होता है, apparently it is a single bond, but it is have a double bond character. So what exactly do we mean by that? So we can say that these are the let's go to the diagram here. So here you can see that this is the particular structure of the polypeptide chain. So as I told you that this oxygen is basically, this nitrogen is having the double bond, having the lone pair of electrons. So these lone pair of electrons will move from here to here, and they can form the another bond, double bond, that is the pi bond. So one bond here is the sigma bond, and other bond is here is the pi bond. So difference between this particular thing is known as the resonance stabilized structure. The actual structure of this molecule, you can say that is somewhere between the peptide bond, single bond, and a double bond. So we say that this resonance stabilized structure, that they have a double bond character. So double bond character basically क्या होता है? That is the length इसकी जो होती है between the carbon and this carbon and the nitrogen atom, that is the between. So let's remember from the organic chemistry that these single bonds basically क्या होते हैं? These single bonds are able to rotate in this space. But this bond having the double bond character, it will restrict the bond rotation. It will prevent the bond rotation it will not be able to rotate the particular this bond in this space. Okay, because we have this double bond character here. So what do we discuss here, that the single bond जो होता है, that is the sigma bond, that is able to rotate, but as this peptide bond having the double bond character, it will not be able to rotate around the space. ठीक है, so this is the double bond character or double bond nature of the peptide bond. Plus this double bond nature of the peptide bond is basically that it will makes the peptide bond planar. All the things, all the atoms in this case will be on the same plane. Okay, so what do we mean by the double bond character? Effects of the double bond will be it's going to prevent its rotation about the peptide bond, and then it's going to make the peptide bond a planar molecule. Now, there are basically two configurations for the amino acids. One is known as the cis configuration and other is known as the trans configuration. Here you can see this is the cis configuration and this is the trans configuration. That is, for this we have to recall our concept of the cis-trans isomerism. Here you can see in trans, the both the carbonyl group, the alpha carbons, they are pointing in the opposite direction. Okay, these are basically pointing in the opposite direction. So here in the cis configuration, both these carbons are pointing in the same direction. So in the trans configuration, so each of the alpha groups, okay, so these two alpha carbon groups, this carbon group and this carbon group, they will never point in the same direction. They will never point it in the same direction. They will always pointing in the different directions. So this is the you can say this is the trans configuration and this is the cis configuration. So normally this trans configuration of the amino acids or in the polypeptide chain, this is thermodynamically it is more stable. It is carrying the low energy level. That's why, because these two groups are pointing in the opposite direction, there will be no bumping of the electrons. There will be no, you can say, when there will no bumping of the atoms, that will there will be no electrostatic repulsion. That will, because of this, it will have the low energy level as compared to the cis configuration. Okay. So, what do we mean by this? If they point away from each other, there will be no steric hindrance. That is, there will be no steric hindrance in case of the trans configuration, but there will be more steric hindrance in case of the cis configuration. What do we mean by the steric hindrance? So, steric hindrance is basically the bumping of the electrons or repulsion of the electrons, as these atoms are basically closer to one another. So these, in case of the trans configuration, there will be no bumping of the electrons. There will be no electrostatic repulsion. So as a result of this bumping of the atoms in this particular case, so there will be no steric hindrance here. But if we discuss about the cis configuration, here there will be bumping of the electrons, there will be bumping of the atoms, and as a result there will be the electrostatic repulsion and there will be the more steric hindrance, and that will carry the more energy. So that's why this trans configuration is basically more stable. It is basically more stable. Thermodynamically it is more, you can say, stable form of the um, polypeptide chain. Okay, now let's discuss about the some torsion angle. So how do we discuss it? That the constant portion of the polypeptide chain, that is it's you can say the backbone of the polypeptide chain. So this is the backbone of the polypeptide chain that we discussed earlier, is the NCC. This is the NCC, that is the repeating unit, that is repeating all over the polypeptide chain. So this repeating unit can be described in terms of the following angles. So the question is, as the protein, this primary structure is basically the linear sequence of the amino acid, but the final form of the protein is in the three-dimensional, you can say, convoluted structure. So how that possible कि अगर हमारी linear configuration में जो peptide bonds हैं that are unable to rotate, then how they will these polypeptide will continue to convolute to form a final 3D structure. So that is the answer, is because of these torsion angles. So these torsion angles are the three. One is the phi angle, that is between the this N and C, this nitrogen of the amino group and the alpha carbon. Okay, so this bond, the angle around this particular bond is the phi angle, and another one is the psi angle. So this particular angle is between is around the bond that is between the carbonyl carbon that is the carbon of the carbonyl group and the alpha carbon. Okay, so these two angles are basically able to rotate at the 360 degree. Okay, and there is another angle, that is the omega, that is unable to rotate at the 360 degree, because the peptide bond is having the double bond characters. ठीक है, तो peptide bond का double bond character है, that basically prevents its rotation. But the rotation is possible in the polypeptide chain. That is because of the phi angles and the psi angles. These phi angles are between the nitrogen of the amino group and the alpha carbon, and psi angle is between the carbon of the carbonyl group and the alpha carbon. So these two angles are basically able to rotate around their axis, so they can rotate about 360 degrees. So this is all about the primary structure of the protein. So in conclusion, what do we say? That the the unique sequence of the amino acid is basically basically determines the actual 3D structure of the proteins. So thank you very much. So that's all about the primary structure of the protein. And if you have any questions, you can ask in the comment section. And if you like the video, then don't forget to subscribe my channel. Thank you very much.